Right, good evening, everybody. Welcome back to my daily lectures. I'm Dr. Bobby. On behalf of New Age Cosmetic White EdTech Health Consultancy, I welcome all of you back to the regular lectures on biology and chemistry. Today we're dealing with the third section on CSIR Life Sciences syllabi on fundamental processes. Let me just read out the syllabus or the portions to be covered in this section. A would be DNA replication, repair, and recombination. Unit of replication, enzymes involved, replication origin and replication fork, fidelity of replication, extrachromosomal replicons, DNA damage, and repair mechanisms. B would be RNA synthesis and processing, transcription factors, and machinery formation of initiation complex, transcription activators and repressors, RNA polymerases, capping, elongation, and termination, RNA processing, RNA editing, splicing, polyadenylation, structure and function of different types of RNA and RNA transport, protein synthesis and processing, ribosome, formation of initiation complex, initiation factors and their regulation, elongation factors, termination, genetic code, amino acylation of tRNA, tRNA identity, amino-acyl tRNA synthetase, translational proofreading, translation inhibitors, post-translational modification of proteins, control of gene expression at transcription and translation level, regulation of phages, virus, prokaryotic and eukaryotic gene expression, role of chromatin in regulating gene expression and gene silence. Right, that being said, we'll move on to the subject proper. Right, we're on to the first section of DNA, and so it's about defining life at the molecular level. We're dealing with the DNA replication and related aspects. The first one would be DNA replication is semi-conservative. Right now, so DNA replication is semi-conservative, right? The elucidation, so this is basically a part of biochemistry too. That's why you see DNA replication chemistry. The elucidation of the structure of the double helix by James Watson and Francis Crick in 1953 provided a hint as to how DNA is copied during the process of DNA replication. Separating the strands of the double helix would provide two templates for the synthesis of new complementary strands, but exactly how new DNA molecules were constructed was still unclear. In one model, semi-conservative replication, the two strands of the double helix separate during DNA replication, and each strand serves as a template from which the new complementary strand is copied. After replication in this model, each double-stranded DNA includes one parental or old strand and one daughter of new strand. There were two competing models also suggested: conservative and dispersive. So you have conservative, semi-conservative, dispersive. Just these diagrams are self-explanatory. Take a screenshot and make your inferences, please. Now, three models of DNA replication in the conservative model. Parental DNA strands, blue, remain associated with one DNA molecule, while another new daughter strands, red, remains associated with newly formed DNA molecules in the semi-conservative model. Parental strand separated and directed in the synthesis of a daughter strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. In the dispersion model, all resulting DNA strands are regions of double-stranded parental DNA and regions are double-stranded daughter DNA. Right, three models of DNA replication in the conservative model. Parental DNA strands, blue, remained associated with one DNA molecule, while new daughter strands, red, remain associated in newly formed DNA molecules. In the semi-conservative model, parental strands separated and directed the synthesis of a daughter strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. The dispersion model, all resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA. Now, scientists Matthew Messelson and Franklin Stell devised an experiment in 1958 to test which of these models correctly represents DNA replication, as shown in figure 9.2. They grew the bacterium Escherichia coli for several generations in a medium containing a heavy isotope of nitrogen, N15N, that was incorporated into nitrogenous bases and eventually into the DNA. This labeled the parental DNA. The E. coli culture was then shifted into a medium containing 14N and allowed to grow for one generation. The cells were harvested and the DNA was isolated. The DNA was separated by ultracentrifugation, during which the DNA formed bands according to its density. DNA growth is N15N will be expected to form a band at a higher density position than that grown in 14N. Messelson and Stoll noted that after one generation of growth in (14N), the single band observed was intermediate in position in between DNA of cells grown exclusively in 15N or 14N. This suggested either semi-conservative or dispersive mode of replication. Some cells were allowed to grow for one more generation in 14N and spun again. The DNA harvested from cells grew for two generations in 14N, formed two bands. One DNA band was at the intermediate position between 15N and 14N. Another corresponded to the band of 14N DNA. These results could only be explained if DNA replicates in a semi-conservative manner. The other two models were ruled out as a result of this experiment. We now know that during DNA replication, models were ruled out. Each of the strands that make up the double helix serves as a template, but from which new strands are copied. The new strand will be complementary to the parental or old strand. The resulting DNA molecules have the same sequence and are divided equally into the daughter cells. This figure Messelson and Stoll experiment with E. coli grown first in heavy nitrogen 15N, then in 14N. N blue band was heavier, that's DNA grown blue band was heavier than DNA grown N red band and sedimented to a lower level of ultracentrifugation. After one round of replication, the DNA sedimented halfway between the 15N and 14N levels. Purple band, ruling out the conservation model of replication. After a second round of replication, this first model of replication was ruled out. This data was supported the semi-conservative replication model. So the question here, think about it, what would have been the conclusion of Messelson and Stoll's experiment if after the first generation they had found two bands of DNA? Right, now its next question will be DNA replication in prokaryotes. Hi, welcome back. Before we could proceed to the further details, let me just explain this diagram here. These are three models of DNA replication. In the conservative model, parental DNA strands, blue, remained associated in one DNA molecule, while new daughter strands, red, remained associated in newly formed DNA molecules. In the semi-conservative model, parental strands separated and directed the synthesis of a daughter strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. In the dispersive model, all resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA, right? Okay, and continuing with the matter proper. Messelson and Stell noted that after one generation of growth in 14N, the single band observed was intermediate in position in between DNA of cells grown exclusively in 15N or 14N. This suggested either a semi-conservative or dispersive mode of replication. Some cells were allowed to grow for one more generation in 49 and spun again. The DNA harvested from cells grown for 2 generations in 49 formed 2 bands. One DNA band was at the intermediate position between 59 and 49, and the other corresponded to the band of 49 DNA. These results could only be explained if DNA replicates in a semi-conservative manner. Therefore, the other 2 models were ruled out. As a result of this experiment, we now know that during DNA replication, each of the 2 strands that make up the double helix serves as a template from which new strands are copied. The new strand will be complementary to the parental or old strand. The resulting DNA molecules have the same sequence and are divided equally into the 2 daughter cells. So I'll just diminish the figures for you to take transcripts, right? Now this figure is about Michelson-Stahl experiment with E. coli grown first in heavy nitrogen 59 and then in 49. So DNA grown in N15 and blue band was heavier than DNA grown N14 and red band, and sedimented to a lower level on ultracentrifugation. After 1 round of replication, the DNA sedimented halfway between the 59 and 49 levels, purple band. Ruling out the conservative model of replication, after 2nd round of replication, the dispersive model of replication was ruled out. These data supported the semi-conservative replication model. Think about it. What would have been the conclusion of Michelson and Stahl's experiment if after the first generation they had found 2 bands of DNA? So here you have a Q&A right at the end of each subject. Keep to it, write it down, figure it out, and post your comments or post your replies, your answers in the comment section. So this is a learning session, so it's not about commenting, it's about giving answers to questions, right? So Q&A would be much respected in this way. Go to it and research as much as possible if you can, because it's the better if you can read more, right? Proceeding to the next, the second part of this subject, DNA replication in prokaryotes. DNA replication has been well studied in bacteria primarily because of the small size of the genome and the mutants that are available. E. coli has 4.6 million base pairs, MBP, in a single circular chromosome, and all of it's replicated in approximately 42 minutes, starting from a single origin of replication and proceeding around the circle bidirectionally, that is in both directions, as you see in the figure. This means that approximately 1,000 nucleotides are added per second. The process is quite rapid and occurs with fewer errors. E. coli has a single origin of replication called ORIC on its 1 chromosome. The origin of replication is approximately 245 base pairs long and rich in adenine thymine sequences. Check out this diagram, note it down, take screenshots, make a copy of it. Replication fork and replication and determinist. Okay, that's about the replication happening. So I'm moving it slowly. So that figure is about prokaryotic DNA replication. Replication of DNA in prokaryotes begins at a single origin of replication, shown in the figure to the left, and proceeds in a bidirectional manner around the circular chromosome until replication is complete. The bidirectional nature of replication creates 2 replication forks that are actively mediating the replication process. The right-hand figure shows a dynamic model of this process. Red and blue dots represent the incorporation of daughter strand nucleotides during the process of replication. Replication overview. The open regions of DNA that are actively undergoing replication are called replication forks. All the proteins involved in DNA replication aggregate as replication forks to form a replication complex called a replisome. Will be there in Table 9.1 and Figure 9.4. So DNA replication in the model organism. E. coli has been extensively studied, providing a foundation for understanding the diverse mechanisms of genome duplication employed by all organisms. In E. coli, DNA replication is initiated at ORIC-C. ORIC-C is melted by the action of the DNA initiator protein to expose two template ssDNA strands that act as platforms for loading the replicative DNA B helicase. One full DNA B hexamer is loaded onto each ssDNA strand with the aid of the helicase loader. DNA C additional exposed ssDNA is quickly coated by the ssDNA binding protein SSB, which protects DNA and blocks additional DNA B helicase loading. Each DNA B hexamer recruits primase DNA G, which synthesizes RNA primers used to initiate DNA synthesis, along with the subunits that comprise the replicative DNA polymerase 3 holoenzyme PO. I think that's 22 or HE. It looks in the it's a helix of L. Okay, you could read it as pole HE. These proteins form the co-replisomes that copy the E. coli genome. Once assembled, replisomes replicate bidirectionally away from ORIC until ideally they undergo programmed disassembly at the termination region, where they encounter sites bound by TIS proteins that create replication fork traps. After completion of DNA replication, the newly synthesized genomes are separated and segregated to daughter cells. Table 9.1. Enzymes involved in the process of DNA replication, the prokaryote E. coli. I'll just read it out for you. E. coli enzyme protein function description. DNA initiator protein melts DNA at ORIC-C, exposing two template ssDNA strands. DNA B helicase unwinds the DNA helix at the front end of each replication fork during replication. DNA C helicase loader loads the DNA B helicase onto the ssDNA strands. DNA E alpha catalyst of alpha catalytic subunit of DNA polymerase 3, catalytic subunit of the main replicated polymerase during DNA replication. I'll just read DNA G. If I've missed out, let me read that portion again for you. It's primase. It synthesizes RNA primers used to initiate DNA synthesis, and then on to DNA Q. Epsilon editing subunit of DNA polymerase 3, editing subunit of the main replicated polymerase during DNA replication. DNA N, beta clamp subunit of DNA polymerase 3, clamping subunit of the main replicated polymerase during DNA replication. PO1A, DNA polymerase 1, processes Okazaki fragments and also fills in gaps during DNA repair process, right? And pole B, DNA polymerase 2, proofreading and editing, especially in lagging strand synthesis, and same involvement in DNA repair. SSB, single-stranded binding proteins, SSB, bind with single-stranded regions of DNA in the replication fork and prevent strands from rejoining. A dimer encoded by GAR A and GAR B. DNA gyrase. Type 2 topoisomerase involved in relieving positive supercoiling tension caused by the action of helicase. A dimer encoded by par C and par E topoisomerase 4, type 4 topoisomerase involved in decatenation of daughter chromosomes during DNA replication. Ligase. DNA ligase fixes nicks in the DNA backbone during DNA replication, DNA damage, and DNA processes. Note, although the genes involved in the formation of the catalytic domain of DNA polymerase 3 are listed. This is the diagram of the origin of replication, all these things together there. Check it out here, the leading strand, the lagging strand, the overall direction of replication with 3 and 5, 3' and 5', the leading strand template, DNA polymerase 3, sliding clamp, single-stranded binding protein helicase, the replication fork, RNA primase, DNA polymerase 3, Okazaki fragment number 2, number 3, number 2, and number 1. Below than the other strand, on 5' you have DNA ligase, DNA polymerase 1, lagging strand discontinuous synthesis, sliding clamp, lagging strand template, RNA primer, and the parental DNA along with the topoisomerase carriage that brings about this, where the DNA ligase is this combination here. Right, the general overview of a DNA replication fork. That's a replication fork that you just saw. So at the origin of replication, topoisomerase 2 relaxes the supercoiled chromosome. 2 replication forks are formed by the opening of the double-stranded DNA at the origin, and helicase separates the DNA strands, which are coated by single-stranded binding proteins to keep the strands separated. DNA replication occurs in both directions. An RNA primer, complementary to the parental strand, is synthesized by RNA primase and is elongated by DNA polymerase 3 through the addition of nucleotide at the 3' OH end. On the leading strand, DNA is synthesized continuously, whereas on the lagging strand, DNA is synthesized in short stretches called Okazaki fragments. RNA primers within the lagging strand are removed by the exonuclease activity of DNA polymerase 1, and the Okazaki fragments are joined by DNA ligase. Right? The primosome assembly. As noted above, the replication of the bacterial chromosome is initiated at ORIC-C, where the initiator protein DNA binds to start the assembly of the enzymatic repliposome machine. The early stages of this process involve the assembly at a primosome that functions to unwind the 2 strands of DNA at the replication forks and add RNA primers to the DNA templates that will be used by the DNA polymerase enzymes to begin replication. Subsequent to the remodeling of the replication origin induced by DNA A, the assembly of the bacterial loader-dependent primosome occurs in discrete steps and involves at least 4 different initiator protein, helicase, helicase loader protein, and primase that act in a coordinated and sequential manner as shown in Table 9.1. The ORIC-C region of prokaryotes contains highly conserved sequence motifs that include an AT-rich box domain that serves as the recognition sequence for the binding of the DNA initiator protein. Initial binding of DNA A to ORIC-C promotes the melting of the DNA double helix and the recruitment of multiple DNA subunits that form a helical oligomer along the newly opened single-stranded DNA, ssDNA. The DNA A protein contains 4 major domains. Domains 3 and 4 are integral to binding the ssDNA, while domain 1 is involved with protein-protein interactions. Domain 2 forms a flexible ligature between the protein interaction domain and the DNA binding domains. DNA melting and helicase loading at ORIC-C. This is the diagram that shows the details. Right, ss9 domain 3 and domain 4, and this is domain 4. And then you have the diagram of the domain 4, domain 3, DNA domain, and the direction of progress, direction of DNA B movement during replication. Right? Now, this is the figure of the assembly of a chromosome DNA melting at RxC and loading the DNA-B0. So this explanation would be over with this lecture, which is this is a shorter lecture, but this is only an intro part. So there's further the larger one, the usual hour-long lecture would be ensuing after this would be continuation after this lecture concludes shortly. So I just finish up this write-up for this lecture, right? And the helicase loader complex onto the DNA bubble. Lower schematic ATP-bound DNA A, the initiator protein binds to the DNA boxes where domain 4, thereby promoting dsDNA to wrap around the DNA filament, causing torsion strain to the dsDNA. Meantime, domain 3 of DNA A binds to one of the two ssDNA strands of DNA, unwinding elements and stretches the strand. These interactions cause the AT-rich DNA unwinding element to melt, forming a bubble. At the same time, binding of DNA C, the helicase holder, traps DNA B, the helicase, in an open lock quasar conformation to enable its loading onto ssDNA. DNA C interacts with DNA A at the end of the filament and serves as an adapter to load one DNA B, DNA C complex. It is not known if closing of DNA B around ssDNA to form a hexameric ring occurs before or concomitantly with dissociation of DNA C. Domain 1 of DNA A interacts with the N-terminal domain of DNA B, helping to load another DNA B, DNA C on the complementary strand. Upper insets, you have the helical filament of DNA A formed by domains 3 light orange and 4 pale green. So there's the colors of the indicators on top. Equifax Helicase, DNA A, PDB, 3R8F and domain 4V, KORAH DNA A, pale green bound to dsDNA, PDB1. The ssDNA binds in the middle of the DNA filament by interactions with AAA plus domain 3 of DNA A. Right, so that's the diagram there. Now a quick look and that should be it, right? And you see the details on it. And the E. coli system, the helicase loader protein, DNA C complex.